Hello, good morning, everyone. Um, let's get started. First of all, welcome everyone to the second MyDog webinar. We are very excited that you can join us here today. Please let me briefly introduce myself. My name is Sui Kwan Tang. I have a PhD degree in environmental microbiology, graduated from University of Toronto. Currently, I'm the chief scientist at MyDog, focusing on developing and optimizing NGS next generation sequencing workflows for infectious disease diagnosis. For those who are not familiar with us, I would give a brief introduction to MyDog. MyDog Animal Diagnostics is an infectious disease diagnostic lab specializing in using NGS, next generation DNA sequencing, to diagnose infectious disease in all animal species in various sample types. Using this molecular-based approach, the MyDog all-in-one test allow clinicians to diagnose any bacterial and fungal infections faster and more accurately than traditional culture testing can offer. MyDog was launched in 2019 and has since then been serving the veterinary community all across the United States, as well as Canada and Europe. Okay, now let's get into the webinar. The scheduled speaker for today was Dr. Janina Kronbeck, who was our director, but unfortunately, Dr. Kronbeck is sick today and therefore unable to conduct a webinar. Instead, another scientist from our team, Dr. Kelly Zapenda, will present the webinar today. We sincerely apologize for the unexpected change in today's speaker. So next, I will introduce today's speaker, Dr. Kelly Zapenda. Dr. Zapenda has a PhD degree and is currently the microbiome scientist of MyDog Animal Diagnostics. She leads the effort to conduct peer-reviewed research on the bacterial and fungal microbiome to establish healthy norms for animals and compare norms with disease conditions in various animal populations. Dr. Zapenda earned her Master of Science and PhD from the University of Southern California, specializing in microbiome research, and has published various peer-reviewed papers on the gut microbiome in clinical settings. She is passionate about furthering a scientific discovery of the healthy microbiome in animal population and translational microbiome science to improve clinical applications. So in order to make today's webinar run smoothly, we will have all participants muted during the presentation, but all questions are welcomed. Please submit the questions to the chat. So there's a button on top says chat. You can click on the button and type in your questions. And we will go over all the questions at the end of the webinar. Okay, now I'm going to pass over to Kelly. Kelly, it's your time.
Thank you, Dr. Tang. So thank you everyone for joining us today. I'm really excited to share some of the exciting work that's come out in the next generation sequencing research in exotic animals. Today we're going to talk about the application of NGS, which is next generation sequencing, as an infectious disease diagnostic tool compared to some of the more traditional diagnostic tools, specifically for exotic animals. So it's pretty apparent that in order for us to have successful treatment of infections, we need to be able to accurately identify pathogens. And for years, veterinarians, clinicians, and scientists have been studying the microorganisms that live in animals and how those microorganisms could potentially impact infectious disease. Collectively, these microorganisms are known as the microbiome. And I would say traditionally, most of the research has been done in small animals like dogs and cats. But of course, we know that bacterial infections are also just as common in exotic animals. And so the microbiome has gained more attention in that area of research as well, specifically looking at exotic animals. We've seen things like salmonella in reptiles and that transferring to their owners, as well as other pathogenic species like Cryptosporidium found in exotic farmed animals. But care for exotic patients can be a little tricky because clinicians often are left to reference small animal data since more research has been transpired in dogs and cats, and or use more traditional methodologies that have limitations and are oftentimes inadequate to accurately identify pathogens. These two obstacles oftentimes lead to less effective antibiotic treatments and potentially less successful resolution of chronic infections. And not only that, but it's important to note that um, obviously bacteria are of main interest when we're talking about infectious diseases, but the microbiome is comprised of many different genetic material, including fungi. However, a lot of the traditional methodologies don't allow for a comprehensive analysis for the fungal microbiome. And we know that particularly with pathogenic infection, infectious fungi play a role as well. This is where NGS-based diagnostics come into play and what I want to be talking about mostly today. So again, NGS stands for Next Generation Sequencing. The testing started in about the 2010s, so it's a pretty newer methodology, and it has a lot of broad benefits that I want to go over right now. So compared to traditional methodologies, NGS allows for the simultaneous detection of multiple potential pathogens that could be contributing to infections. You're able to look at all aerobic and anaerobic bacteria as well as fungal species with their cell counts. So you're not only able to identify a particular pathogen, but you could look at the concentration of that pathogen to see if it's actually impacting the disease. NGS also offers a high degree of sensitivity and specificity for the microbial profile at the site of infection. You can look at multiple sites and you can also get sort of a snapshot at the exact time of collection as well. And NGS offers a unique diagnostic tool where you're able to look at antibiotic resistance panels, even for more novel pathogens or pathogens that are not able to be cultured or tested with traditional methodologies. And because of these broad benefits, we're able to confidently detect actual pathogens that could be causing any infection. Now, I want to go over how NGS works, how we're able to identify these pathogens. So let's just go over the workflow briefly. So if you get a specimen, you can swab that specimen. You can swab any site. Let's say you swab the skin. From that swab, you'll get a lot of different genetic material. You'll get rods, cones, anaerobes, aerobes, spores, you name it. You put that into a stabilizing solution, which you see in this tube kind of looking thing right here. And that tube has a stabilizing solution in it, meaning that we can kind of get a snapshot of what the bacteria look like at the exact time of collection, as opposed to keeping it at ambient temperatures, because of course bacteria are pretty temperamental and fungi are as well. And so if the temperature changes, then certain bacteria or fungi can degrade or maybe grow in excess or artificially. Once we have that sample, then we're able to extract DNA, and this is sort of the core of NGS technologies. So we extract DNA and we're able to look at bacteria or fungi and essentially look at the entire genomic sequence of those bacteria or fungi and then classify them. There's a couple of ways that you can extract DNA. You can either look at the entire genomic sequence. Oftentimes this tends to be a little bit more expensive and it also is very time consuming. And so a very common way to sort of get around those obstacles is to do something called looking just at a portion of the DNA sequence. When we're looking at bacteria, we'll typically look at the 16S region of that bacteria. So it's only looking at one region, although the 16S region is included in virtually every bacteria. So when we look at this portion, we can be confident that we are actually detecting the bacteria that is present. And similarly, ITS is makes it possible for us to look at that in fungi. We can take these fragmented pieces and we can generate millions of DNA sequences. And what we'll get is a whole bunch of sequences, like millions and billions of them. And from that, we're able to detect all pathogens in a particular specimen. So we'll get a lot of different sequences and different iterations of ATCG, like any gene. And what we can look from that is what bacterial type it is and how many counts there are. And the same thing goes for fungi. Once we have all of this data, then we can look at the comprehensive microbiome. So let's go over what the microbiome is briefly. The moment our pets are born, they're being colonized, and they're being colonized by things like bacteria, protozoa, fungi, archaea, as well as viruses. And collectively, all of this material is known as the microbiome, which essentially means small life. There are many different factors that impact the microbiome of our pets: how they were born, their DNA, their epigenetics, age, diet, medication, environment, lots of different things. In order for a microbiome to be classified as, quote, healthy, there's a couple of different measures that we look for as microbiome scientists. So what you see here is sort of a donut chart, and you can see a lot of different colors. Each of those colors represents a particular bacteria. And it looks like there's quite a bit of colors, which means that there are quite a bit of bacterial types present. This can be measured as diversity. So the more bacterial types present, typically the higher the diversity and the healthier the microbiome is. But what we also want to see is that among these bacterial types that it's balanced. And so we want to see symbiosis, meaning that there are enough commensal or healthy bacteria or fungi present and that there are little to no pathogens present. Conversely, if we look at a sample that's infected, what you see in this donut chart on the right is clearly less colors, meaning less bacteria are present. And what you also see is an overrepresentation of certain bacteria. In this case, there's an overrepresentation of Streptococcus in orange and Pseudomonas in blue. Both of these are typically pathogens, depending on the species type, of course. And so what you see is that there's this imbalance or dysbiosis in the microbiome compared to the left side, which looks a little bit more balanced. What we typically look for in a diseased population or in an infectious sample is more pathogens and biofilm. And then we also look for, are these particular bacteria corresponding directly to an infectious disease, which is known as a pathobiome. At MyDog, we do this type of testing all the time, where we'll compare different groups of animals. We'll look at a healthy group or an asymptomatic group, and we'll compare them to a particular infection or pathogenic disease. I wanted to highlight a couple of these studies that we've published in the past few years, although we have many more studies on our website, and I'll have a QR code at the end of the presentation if you want to check those out. So for instance, we recently published a paper in 2022 where we compared bunnies who were asymptomatic to bunnies who had otitis or inflamed ears. And you can see with these donut charts, we obviously don't need to go into the specific bacteria, but you can see that there's significant differences between the types of bacteria that are present as well as the concentration. And similarly, in another study that's about to be published by our group, we compared asymptomatic ferrets to ferrets who were suffering from diarrhea. And you can see the diarrhea group has less bacterial types present, as well as an overabundance of these pathogens compared to the asymptomatic group. Now, how does this translate to the clinic? Well, we also work with clinicians who they'll send us one sample or a few samples of their patients that are suffering from various diseases, and we can help them to sort of direct their treatment with our with NGS technology. So what you see here as an example is Mystique. She's a bearded dragon who was suffering from yellow fungal disease and facial lesions, which you can see in this picture here. Their vet ordered a microbiome testing report, and that helped them to direct their antibiotic treatment to administer. And fortunately, after 1 month, the lesions were significantly improved. But of course, MyDog isn't the only group that's producing microbiome research in exotic animals, which is great because it definitely takes a village to, bolster this research up. There are a number of key players in the exotic animal research space that have produced work alongside of us and really have allowed us to, sort of expand on this diagnostic tool. There's tons of research in populations like lemurs, in koalas, in pretty much anything, lizards, monkeys, rabbits, let's see. Sloths, if I can read, also amphibians and beetles and even birds. And you might even see some familiar faces in these authors here. There are a lot of key players that have really dedicated their entire research career, to better understanding the exotic microbiome. People like Dr. Robert Dalhousen, which is one of the leading experts in exotic animal microbiome research. He actually gave MyDog some of our first exotic samples and really encouraged our group to expand this area of research further. With this expansion of this research, there's sort of a light at the end of the tunnel where we understand the exotic microbiome better, but there are still some gaps in the literature and some things that we need to address, and NGS technologies have the capabilities to do just that. The two main areas that need to be addressed, the gaps in the literature, are the lack of fungal analyses and also a need for us to consider something called antimicrobial resistance, or AMR. Mainly the reason why there are these missing links in exotic animal research in particular is because of the limitations of the traditional infectious disease diagnostic tests. So one of the most common kind of long-standing traditional diagnostic tests is culturing. This has been around since the 1800s. It's used quite often in clinics, but it has quite a few limitations. First and foremost, there are transit changes in the microbial profile that occur with this type of testing. Culture testing, it's often difficult to look at anaerobes, anaerobic bacteria specifically. And it's difficult to look at multiple tests, or you need multiple tests to target different pathogens versus being able to target different pathogens in one test. The turnaround time for getting the results for these tests is often pretty long. So it takes about 2 to 7 days to get results back for bacteria and about weeks for fungi, if at all, because only 3% of microbes are culturable. And we know that there are many different microbes that can impact pathogenic diseases. Of the 3 that are culturable, about 60% of the tests, oftentimes you get no growth, so you don't really get any clarity on which bacterial pathogens are causing a particular disease. There's high sensitivity with culture, but only for culturable pathogens. So if you're constantly culturing the same pathogens, you might be missing out on some novel or less known pathogens that could be contributing to a particular disease. And with culture testing, you're unable to look at biofilms, which are the cause of 80% of infections. So NGS is typically better than culture testing because you can look at more pathogens, you can look at them simultaneously, and you can get the results faster. Another common traditional disease diagnostic test is a PCR test, or qPCR, which was developed about a century later in the 1980s. This test has high sensitivity and specificity, which is great for clinically diagnosing things. However, you can only look at a few selected targets at a time, typically about 5 to 8 pathogens, and you really have to have a good educated guess of which pathogens you think are causing a particular disease. So you could be missing out on some novel or unknown pathogens. The panels often return negative, and there's a high false positive rate, which can lead to misdiagnosis. And you're unable to look at which particular, if a patient has a certain antimicrobial resistance, it's limited, or you're not able to look at it at all. And looking at cell counts can be pretty difficult with this type of test. So NGS is superior to PCR testing because you're able to look at new or novel pathogens, and you can look at multiple pathogens simultaneously versus having to pre-select 5 to 8 different tests or different pathogens. So it seems like NGS should be the new standard of care, but the question is that true? And so our group actually published a study recently earlier this year, where we wanted to compare these traditional tests to NGS to see if in fact it is superior. So we took a group of bunnies who had ear inflammation, and we did a culture test and also NGS. We only tested bacteria and fungi that we could culture as well as detect with an NGS method. And what we found is that you can see on the left pie chart is the bacteria and on the right is the fungi. What we found, if in the dark blue, you can see that 14% of bacteria we were able to detect via NGS, but we weren't able to detect via culture. So the culturing missed 14% of the bacteria. And in fungi, it was even greater. There were 49% of fungi that were not detectable via culture, and there were also 24% that no fungal culture could be done. So there was about 74% of fungi that were missing when we're looking at cultures versus looking at NGS testing. And this just gives you an example of, some of the common bacterial infection or bacterial pathogens that we were able to detect with NGS that we weren't able to detect as well with culture, and some of the infectious fungal species. So the other issue is that typically if an animal has a bacterial infection, the best course of action is an antibiotic. However, many people and animals alike, there's a global issue of AMR, or antimicrobial resistance. So when antibiotics were developed in the 1940s, they became really effective, but in the 1960s and into the 70s and now today, this effectiveness has reduced. And that's basically because the development of new classes of antibiotics is far behind the need for antibiotics in our population, in our world. And this is likely because there has been antibiotic resistance that has evolved with these different bacterial pathogens. And since millions of animals suffer from multifactorial infections every year, we really need to address AMRs when we're treating our animals. And it's so much of an issue that it's purported that by 2050, about 10 million deaths will be because of AMR, and it will actually be the leading cause of death. Fortunately, we at MyDog and many other research collaborators have been using NGS to identify different AMRs to help bypass this issue. I'm not going to go over all of these studies, but this is just kind of a, to give you an indication of what has been done in the past. And of course, MyDog is a great resource for this. It's one of the things that we do pretty standardly. And aside from antibiotic treatment, we can also use NGS to identify other types of treatments that we can implement with our patients as well. Things like probiotics, which have been shown to positively modulate the microbiome, as well as prebiotics, which have been shown to help feed healthy microbes and increase commensal bacteria and reduce pathogens. So I want to go over the workflow that we do at MyDog, which allows us to obtain this information from NGS testing in a quick and timely manner. So we have a very simple collection kit at our that we use. We can use it on pretty much all animal species. We test dogs, cats, bunnies, animals, or birds, reptiles, and we can do pretty much all sample types. So we can look at nasal microbiome, the skin, urinary tract, the GI tract, ear. Pretty much if it fits into our sample tube, then we can test it. And then we do all of our wet lab and bioinformatic analyses in-house. So this allows for us to go from start to finish, from getting the sample of the bacterial fungal pathogens all the way to generating the reports in a really short period of time, typically a 48-hour period, which is great for clinicians because if you're needing to treat your patients, of course, the quicker you can get the results, the better. The other nice thing is that these diagnosis reports are very accurate. We have high specificity and sensitivity. We're able to identify many different bacteria and fungi and pathogens, and we have a tailored antimicrobial therapy that we can get because we can look at 43 different antibiotic resistances to antibiotics, particularly for each patient. And we have 3 different types of kits. So you can see in the yellow, we have a urine kit. It's specifically for urine. And then the 2 green kits, we have a smaller swab and a larger swab, depending on the sample site. And then we're able to generate comprehensive reports for our clinicians. So I want to go over a couple of 3 different case studies that kind of show how NGS and how MyDog technologies can be helpful for different animals. So the first animal that we have is a whale that was experiencing anorexia, nausea, and lethargy. And this was purported to be because of a GI infection, which pilot whales in particular are at an increased risk for. So we worked with a group to administer a treatment called a fecal material transplant, or FMT, which is essentially used to repopulate the fecal microbiome and treat these GI diseases. Our collaborators used MyDog's NGS testing to test the FMT treatment in a couple of different ways. We first used the NGS testing to identify which bacteria and fungal taxa populated the whale microbiome. We were able to use the testing to identify which whale FMT donor would be most appropriate for our patient. And then we were able to track the whale's microbial progress after they received the FMT treatment. And what we found is that FMT increased that common indicator of gut health that I mentioned previously, bacterial diversity. And by us tracking the microbiome over a period of time, we were able to help the clinician administer effective treatments to alleviate the anorexia and nausea and lethargy that they were experiencing. A second case study was we worked with a 29-year-old Congo African gray parrot who was suffering from soft malodorous stool or stinky stool. And this is a paper that was actually just recently published with us and a collaborator. Additionally, this parrot was, the clinician did culture testing with this parrot to try to find particular pathogens that could be contributing to their issue. But the problem with the testing is that they were able to identify 3 nonspecific potential pathogens without actually knowing what the specific pathogenic species was that was contributing or overabundant in this African gray parrot species. This is where the MyDog testing or NDS testing came into play. We performed DNA sequencing or MDS, which is essentially next generation sequencing, to identify the specific bacteria that could have contributed to this disease. We found 3 that contributed, and they were very specific species at the species level. And because of this, the clinician was able to produce, a faster, more meaningful diagnosis and treatment, and the gray parrot was able to have their disease resolved. And then the last case study I want to go over is with a 7-year-old cake bird who had severe anemia, less than 30% hematocrit, due in part to intermittent and severe bloody diarrhea. So this was similar before they used the NGS technologies. The clinician treated them with what they called a random approach, where they gave them antibiotics and anti-inflammatories that they thought would be beneficial. And this treatment helped the bird to improve, but once the antibiotic treatments were stopped, then it returned. When they came back for a returning visit, they found that the anemia had gotten even worse, that their hematocrit levels had gotten down below 20%. And so they decided to run a bunch of tests. They did a blood transfusion, they did fecal diagnostics, cytology, culture, and MyDog's NGS testing. The bacterial cultures came back negative. The cytology showed small numbers of concerning yeast-like organisms found, but they didn't know what the specific organisms were, and then the fungal culture came back negative. But when they ran the MyDog NGS test, they found 4 bacterial pathogens that could be contributing to this bloody diarrhea, and they also found an infectious fungi that was shown to be contributing to the disease as well. And so because of this, they were able to administer an antifungal and antibiotic treatment for 2 weeks. And fortunately, the bloody diarrhea resolved. So in conclusion, NGS offers superior pathogen detection, particularly for specific bacteria like strict and facultative anaerobic bacteria. We can look at slow-growing bacteria as well as gram-positive bacteria and novel and rare pathogens that we would otherwise not be able to look at with the culturing or PCR testing. In addition, NGS allows us to look at all different fungi that could be contributing to infectious diseases and allows us to test for antimicrobial resistances or AMR. So it really does have tremendous potential as the new standard of care for exotic and zoo animals, which is really great news. Thank you so much. And if you'd like to learn more about what we do at MyDog, our director, Dr. Crumbeck, was recently on a podcast, and you can use the QR code to check that out. And then you can also check out our website. Thank you very much.
Okay. Yeah, thank you, Katie, for the great presentation. So next, let me see. I do connect some questions from the chat. So let's go over the question together. So the first question is from Dr. Thomas DeMar. I think he was asking about, considering that marine microbiology is an understudied realm, what advice would you give for submission of sea turtle specimens?
That's a good question. Yeah, I believe we test marine animals. I don't know specifically about sea turtle specimens, but I think the best thing to do is to get as many samples as possible. because the more samples you can get in a particular population, then you can kind of use those samples as a reference. So then if you're looking at an animal that might have an infectious disease, then you can compare it with that. And that's kind of what we do at My Dog, where we'll try to get as many healthy animals as possible in a particular population, so that when we get a bunny, for instance, we'll have a big group of healthy bunnies, and then if we get a bunny that has an infection, then we can sort of compare the microbiome of both of those. So it's really just about getting the samples. But the nice thing about the NGS test is that we're still able to reference, even like, let's say you're going to be testing a sea turtle, and you're not necessarily sure of their healthy microbiome profile. You can look at certain pathogens that have been shown to be pathogens in other sea animals, or are typically pathogens just across the board. So you can kind of sort of direct your treatment in that way.
Okay, great. The second question, it's about, it was asking if the recording will be available and where can we find it. That's after the presentation. I think I can answer that question. So this webinar will be recorded, and then the recording will be made available after this webinar to all the attendees, and also including the people who are signing up for this. Yeah. The third question, which is a very common question, I think we normally get from exotic animals as well, and that was, can a mycoplasma be detected using the MyDog NGS testing?
Yes, it can be. And in fact, I just worked on a project in bunnies where we found mycoplasma was present in that population. So yeah, it's definitely, it's an important pathogen to look at for sure.
Okay, the next question here is, with more research funding, bacteria and viral co-infection in exotics, is there a possibility to include virus in NGS in the future?
Oh, that's a great question. I'm not sure. Dr. Tang, do you think there is? Can you give some insight?
I think so. So currently the MyDog NGS sequencing workflow is more targeted, as like Katie described earlier, it's more a targeted based approach. So currently we do not include viruses. But NGS, if you're using more shotgun metagenomic approach, covering virus is possible. Yeah, so the answer is yes. And we actually are working, MyDog is also kind of continuing following the advance of the technology development as well. So we are looking into other approach to continue improve our what we offer for NGS testing. So yeah, I think that's quite possible. Okay, the next question. Do you have the ability to speak a case over with a DVM to clarify normal versus pathogenic organisms? I think the question is asking, do we have a vet that was maybe available to help clarify to help explain the report? I think that's a question.
Yes, yeah. So we have a couple of collaborators, DVM collaborators, that we can kind of reference out to if need be. So like if you get a certain report and you're unclear of a particular pathogen, you want to talk it over with one of them, you can. And then you can also talk to myself or Dr. Krumbeck, who were very familiar with different pathogens in microbiology. So you can talk to both or either of us depending on the case.
Okay, I have another question here. How does the results report is presented for the clinician to interpret? Is there any support reachable by phone for assistant to interpret pathogenic versus normal flora?
Yeah, so I think that kind of goes into the previous question as well. So the reports are very user friendly, I would say. They're extensive. There's a lot of data. And so we definitely will walk through that data with you so that we make sure that you understand and are able to interpret the reports. I'd say that they're as user friendly as they can be for microbiology. It can get a little dense for sure, but we're always available via phone or to interpret and kind of talk through certain pathogens or normal flora. And I think one of the questions we get a lot is, sometimes there's pathogens that are present in a sample, but it's at a really low concentration. And so then the question is, well, is that pathogen actually impacting the patient's disease or not? If it's at a really small concentration, we can also kind of talk you through that. And we're familiar with, for the most part, we're familiar with cell counts and how those concentrations impact the disease, and can kind of walk you through that as well.
Okay. Another question, are there similar NGS technology companies being applied in human medicine?
Yeah, so we can use NGS for any sample, and NGS is widely utilized in human microbiome research as well.
Okay, I think that's all the questions we have right now. Okay, I think that's it. Presentation for today. We would like to thank everyone again for attending this webinar, and just let's keep in touch. And if you guys have any questions, feel free to go to our website, www.mydoctest.com. And there's a lot of information and resources available on our website, including case studies, including example reports. Yeah, again, as the website is www.mydoctest.com. You can also email us at info@mydoctest.com. info@mydoctest.com. Send us an email when you have any questions. Okay, I would thank you very much for attending this. We will see you next time. Okay,
thank you.
Bye.